Good afternoon, everyone. Thanks for joining us this afternoon to talk about forecasting the spread of viruses in swine in the United States. I'm excited today to share with you some of our ongoing research on translating movement and environmental data to actionable information to better control infectious diseases in this population. The disease that we're going to be focused on today is called porcine epidemic diarrhea virus, which is a alpha coronavirus. It's not usually considered among the fast diseases, but it shares a number of characteristics with fast diseases. It is rapidly transmittable, has large economic impacts, and it does regularly jump international borders. For example, in 2013, PED was first detected in the US, after which it rapidly spread throughout the country and impacted approximately 30% of breeding farms within approximately 1 year. And the primary impact of this disease is that it has a very high mortality rate for neonatal piglets. So this initial incursion of the virus in the US caused the death of at least 7 million piglets and an overall depression in pork production for that year. So in the United States, we lack a regulatory framework for monitoring endemic viruses in swine. And this is true for PED, and it's also true for a number of other viruses like PRRS. And so to fill this void, the University of Minnesota began the Morrison Swine Health Monitoring Project approximately a decade ago. And this project is based on close industry partnerships and participation. And currently we have around 50% of the US sow population enrolled in the program, which means that they send to us on a weekly basis which of their sow farms, or the infection status of their sow farms. So from that, we can start to really be able to build an epidemiological understanding of the trends of the disease, as well as we use this data for research. So this is the type of data that we produce. This is the plot for PED with the red color showing the farms that are actively infected with PED through time. So you can see that early initial epidemic that occurred, that we're around 30% of the farms were infected. And then you can see that there's an ongoing endemic state of the virus in the population. So we wanted to, in this project, ask, can we use this very rich source of retrospective data to actually predict future risk? And the way, before we get into how we predict risk, we need to talk a little bit about mechanisms for between farm spread. It is for PED, as with many viruses, animal movements is one of the key factors in spread. In the US, pig production is primarily vertically integrated multi-site production. So there is an enormous amount of pig movements within the country. The other primary route of spread is local area spread. So this is based on spatial proximity between farms. This includes mechanisms like airborne spread, but also any other transmission mode where the probability of transmission is going to scale with spatial proximity between farms. Other important components of spread, but maybe are less important, are contaminated equipment in vehicles that carry fomites, as well as contaminated feed, which I think was more of an important mechanism early on in the epidemic and less so now. So because we can primarily think about risk then as in terms of spatially independent processes related to animal movements, as well as spatially dependent processes related to spatial proximity between farms. So we want to develop an approach that would actually allow us to develop machine learning models based on a neighborhood-based approach. And this would allow us to couple local and long-distance processes and transmission. So what I mean by a neighborhood-based approach is that we have a sow farm of interest on a landscape, and we draw a circle around that sow farm of a 5-kilometer radius, and we consider that the neighborhood of the sow farm. And we have information about farms in that neighborhood in terms of where are they, as well as if they are a sow farm, we may have their infection status. And then on top of that, we have our animal movement data. We consider this in two ways. One, the movements of animals into the focal farm itself, but then we are also summarize the movements that are being made into neighboring farms of the focal farm. So for example, if we know that this neighboring farm maybe moved in pigs from a PED positive site. And anytime we're moving pigs into the neighborhood, that increases the likelihood of a local introduction of the virus. And then from there, the virus may spread through to our focal sow farm based on spatial proximity, which we expect that various microenvironmental factors would modify the likelihood of that transmission to happen. So these things include land cover, vegetation, temperature, etc. So this time lapse is basically a weekly time lapse of the infection status of sow farms. So we can see these different sow farm neighborhoods, and then we can also see the flow of pigs between those neighborhoods, which is not shown in a dynamic fashion here. But essentially, we feed our data on infection status, pig movements, and a number of our environmental and spatial variables into a machine learning algorithm to try to predict weekly infection status. And we use a machine learning algorithm called XGBoost to do this. Just to talk a little bit more about the outcome that we are trying to predict. We are trying to predict weekly outbreak risk, but we are trying to predict that not for the current week, but actually 2 weeks in the future. So the data that we're using to make these predictions is actually lag data, because we don't actually have the data for the coming week. But the fact is if we want information to be actionable to our partnering producers, we actually need to be able to give them some information early enough that they actually are able to respond to this information in some sort of mitigation or prevention step. So we use a total of 6 weeks of movement data, including 5 weeks of retrospective data, as well as 1 week of the scheduled movements. And then we use this information combined with our infection status and environmental data to actually make the prediction about the following week, or 2 weeks in advance. One of the types of outputs that we get from this analysis is a variable importance plot, which is essentially a ranked list of how important each factor was in predicting the outcome. And what you'll notice is right at the top of our list is the PEDV status of the farm of origin of any movement that's made into the focal farm, which makes sense. If you're moving positive pigs if you're moving pigs from a positive site into your farm, then there's a good chance you may have an outbreak as well. But interestingly, we also find that the PEDV status of the origin farms for the movements made by neighbors also was an important predictor of outbreak risk for the sow farm, which is an interesting finding in itself. Our model has a sensitivity of 20%, as well as a positive predictive value of 70%. And you might say, well, 20% is not very good. But when we talk to our partners, they say, well, this is more information than we had before. And the fact of the matter is if you try to increase sensitivity because of the structure of the data, if you increase sensitivity, you'll decrease the positive predictive value. So you may flag more outbreaks, but you also have more false alarms. And our partners indicate that they would rather have fewer false alarms and that have still missed some of the outbreaks, but have that information be truly actionable when they receive it. So this is how we ended up with this balance of positive predictive value and sensitivity, and it is something that we will continue to work on in the future to improve that. So our partners currently receive a report from us on a weekly basis. They receive a list of all of their farms and the associated outbreak probability of those farms. We also send them a kind of line plot that just shows the trends in risk through time. And then we can visualize this risk spatially as well to see if there are any hot spots. It's incredibly important for us to think about the sustainability of this project. So we have developed an automated pipeline to actually facilitate running this analysis on a weekly basis. And this is also why we really focus on data that's already routinely collected. So animal movement data, for example. There are a number of other things we could certainly consider adding, but if it becomes too much for farmers to really keep up with the data requirements, then the system just doesn't have the long-term viability to really keep people engaged. So that's why we keep it simple and focus on data that's already available. And then we also have thought ahead to the scalability of this project. And one aspect of that is that we've actually, within our automated pipeline, we've built in a model updating feature that allows the model to adapt to evolving epidemiological contexts. An important benefit of this is that it will also allow us to be able to expand this pipeline to a new disease. So, for example, an African swine fever or foot and mouth disease or something else like that that could come into the country. As long as the data is managed in the same way and we can put the data into the same structure, we could run it through our pipeline and produce forecasts through our pipeline. So in conclusion, we've developed a forecasting platform that allows us to combine animal movement data, spatially and environmental data, as well as outbreak data to actually forecast the probability that farms will break 2 weeks into the future. So this provides us to provide advanced 2-week warning of which sow farms are most at risk, which is then information that we can share back to the producers, and then ultimately they can use it for decision making and to prevent or mitigate the impact of the disease. I just want to briefly recognize the two main people that are part of making this work happen: Igor Poplawski, who's a postdoc in my lab, as well as Rahul Bhojwani, who is the data science backstop that did much of the work for this project. And thank you for your time.